Just like let you know. Just think now. Don't worry about it. I'll give you ideas.
You'll see me like freak out and grab my pen. Okay.
Yeah, see, exactly right.
Okay. Hi, everyone. I'm Gina, for those of you who don't know me. Thanks for coming to Dr. Emily Joseph's seminar. I'm super excited to get to introduce you to her. She is a current assistant professor at MSU in the plant biology department. And she comes to MSU originally. She started her undergraduate or worked as an undergraduate at Brown University with Annie Schmidt, working on phenotypic plasticity, and working as a technician, right? No. Okay. And then went on to graduate school at University of Toronto, working with John Stinchcombe and Stephen Wright. Then went on to UC Davis as an NSF postdoctoral fellow, working with Graham Coop and Jeff Ross Ibarra. And I am super excited that she is here, going to present a talk to you today about the evolution of plasticity, or G by E interactions. And hopefully we will have her. She works on a lot of different species, and I'm trying to convert her to Morning Glories, which I think is working. And all right, go ahead.
Thank you, Gina, and thank you everyone for coming. It's really fun to get to talk to you. I feel like in the time that I've been at MSU, it's been really great to have UM nearby because it's such a cool department, so many great scientists here, so it's fun to get to visit formally, but I've really enjoyed informally having you all down the road. So thank you for coming to my talk. And in particular, having Gina nearby has been awesome. She's been such a great unofficial mentor as I've been a new assistant professor. Okay, so the big question that I'm interested in is, why is there genetic variation for traits within species? I don't know why species is blue, but that's not, I don't want there to be genetic variation within my title, but so I'm an evolutionary biologist, I do evolutionary genetics, and so the questions that really motivate me boil down to when we look within nature, why do we see variation within species, whether it's for things like life history, timing, color, size, defense traits, or anything else. We constantly see this variation, and I want to know why that's the case. And so I tend to think about this through a couple of different types of hypotheses, and so I'll move away from pretty plants and talk to you about cartoon plants, and thinking about what evolutionary forces are shaping genetic variation for traits. And we often think about this in contrast to the idea that at any given place and location and time, there might be some optimum trait value. So in this case, I'm illustrating that with this in plant size. You might imagine that there's some optimum size for our cartoon plants, and selection then we expect to move plants and populations towards that optimum and eliminate variation. So why is this not what happens everywhere all the time? So the first explanation might be that traits evolve neutrally. Maybe we see variation because there's no optimum for the trait we're looking at. We're just seeing mutations happen. They create variation. They don't affect fitness. And that's why we see a lot of variation in nature. And so that's always one potential hypothesis. Our next hypothesis might be that mutation isn't strong enough to balance selection. So we always have selection moving things towards an optimum, but mutation happens often enough and is strong enough that we see this variation that persists at a balance between mutation and selection. And so this means that a lot of the variation that we see might be somewhat deleterious, but it's not so deleterious that it's getting eliminated right away from the population. So this would create variants that are under negative selection and a general model of stabilizing selection. Our third hypothesis is that balancing selection maintains variation. And balancing selection is a catch-all term that could include a lot of different processes, like There might be differences in optimum year to year. We call that temporally variable selection. It might be that the rare trait is somehow favored, whatever is rare is more favored, or anything else like that. There's a special type of balancing selection called local adaptation, where the optimum varies in space, and so we end up seeing variation because the optimum trait is different in different places. And so I just wanted to walk through like our main evolutionary genetics hypotheses for why we might see variation within species. Okay. In particular, most of what I'll talk about today also includes thinking about what role the environment plays. And so when we look at plants in our cartoon landscape, they all are growing in an environmental context. So in a really cartoon example, we could imagine that if plants are growing in a rainy environment, they might get to be bigger than if plants are growing in a dry environment. And this difference, if these plants are genetically identical, is often called phenotypic plasticity or an environmental variation. And so I'm really interested in thinking about how we can incorporate ideas about plasticity into our evolutionary genetics models for what maintains variation. And so I'll just argue that I think plasticity is really key for understanding trait evolution. And that is because every trait we look at is in an environment, whether we're looking at things in nature or on a cartoon plants on a slide. The phenotypes that we see are being shaped by the environment. That means that how genes create phenotype is affected by the environment. That affects how selection then acts on our phenotypes and works down to affect how selection shapes genes. So the entire process of maintenance of variation is really hard to disentangle from the environment. And so that really big picture is what I'm interested in my lab, and I'll be talking about projects related to that today. But I first just wanted to go on a little digression to talk about why I think it's been hard to think about plasticity in an evolutionary genetics context. And that is just, it's really confusing, and there's a lot of confusion, which leads to arguing in the literature about plasticity. So one of the big things that's confusing about plasticity, first of all, I think is the distinction between active versus passive plasticity. Sometimes people, when they talk about plasticity, they only mean active plasticity. So that would be the idea like these acacia plants make thorns in response to herbivory. The plant is somehow making a choice to create a trait in response to its environment. Versus passive plasticity might be this plant is smaller because it got eaten, and now it has fewer resources to grow. And I think there's a lot of reasons to be interested in active versus passive plasticity because the mechanisms are really different. But the end result is that the plant or the organism is now different because it's been in a different environment. So for what I'm talking about today, that distinction is less important. People also often get confused about the idea of whether plasticity is beneficial or not. So we could think about adaptive plasticity, like in the previous example. If you're an acacia plant and getting eaten, it's probably adaptive to grow thorns, and people have done experiments to show that. But plastic responses to the environments might not be always beneficial. My undergrad advisor, Annie Schmidt, once proclaimed that death is plasticity. So, just like these dead plants. And I think that's true. If we think about environmental responses, some of your responses to the environment might be maladaptive and are often likely maladaptive. And finally, measuring plasticity can be really, really complicated, and people have measured it in a lot of different ways. So I've just included this schematic to talk about, two ways people often measure plasticity. So here on the x-axis, we have different environments, environment A and B. We have two different genotypes measured by those lines. The y-axis is trait value, and genotype 1 is showing this strong environmental response, or phenotypic plasticity. Genotype 2 has no response. We can also think about that same situation if we think about our different genotypes on the y-axis, and now we're thinking about our environments. In environment B, we see a lot of genetic variation for traits. In environment A, we don't. And we can think about, in this case, it's sometimes easier to think about the fact that genotype 1 is carrying alleles that increase the trait in environment B, but not environment A, whereas genotype, all of its alleles are somehow combining to have the same effect across environment. And so I could probably give a whole talk on different ways to measure plasticity. I will not do that today, but it can be really complicated, and that leads to people having different conclusions from their data. Okay, so today I'm going to talk about 3 different stories from my lab thinking about plasticity in an evolutionary genetics context. The first will be thinking about whether gene expression plasticity is beneficial. The second, trying to understand why organisms aren't perfectly plastic. And the third is an in-progress project from my lab looking at genetic variation for plasticity in an urban plant. Okay, so as I mentioned before, environmental responses or plasticity can often be really adaptive. A classic example of this is the shade avoidance pathway in plants, where when plants are grown in a sunny environment, they might have this growth form, and in contrast, the shade avoidance pathway, plants, when they're grown in the shade, will have longer stem, like an elongated stem and elongated leaves. And the idea is that if you're in a plant surrounded by other plants that are a little bit bigger than you, growing really fast up high is a good strategy to try to capture sunlight, and so this can be adaptive. However, this response could also be maladaptive. For example, if the shade you're under is being caused by a giant tree, it doesn't matter how tall you grow, you're never going to grow taller than the big tree shading over you. And so a lot of herbs that live and plants that live in the forest understory have lost the shade of response pathway. Okay, we know that plants, this response, the shade response can be adaptive because of some really clever experiments done by Susan Dudley and Annie Schmidt, where they took impatiens plants, they induced the shade response versus the sun response when the plants were young, and then they transplanted them into different environments. So high density would be shaded, low density is sunny, and then this y-axis is fitness. And what they found was that, if you induce the shade response to make the plants elongated, they have higher fitness than the sun plants in the shady environment. In the sunny environment, the shade response reduces fitness. And so this is a really classic example of how that shade avoidance pathway is adaptive. However, for a lot of plastic traits and plastic responses, they're not as amenable to these kinds of experiments. You can't just induce it and then have the plant hang on to that response for the rest of its life cycle. And so one thing I've always been interested in is how we could answer questions about whether plasticity is adaptive or maladaptive in using other types of approaches. And so conveniently for me, I started talking to Gina about these questions, and she and Megan Van Etten had done this really cool experiment looking at pesticide resistance in Morning Glory and how treating Morning Glories with herbicide affects expression responses in plants. And it set up sort of the perfect data for asking questions about adaptive and maladaptive plasticity. And so you guys hopefully are familiar with the system because Gina is here. All these plants were grown somewhere nearby. But I'll tell you about the project. So Gina and Megan started with Morning Glories that had been collected from somewhere in Georgia. They did 3 generations of artificial selection. In some of them, they treated them with glyphosate, which is the main ingredient in Roundup, and selected for plants that were resistant to glyphosate. They also had replicates where they did the same selective processes, but they didn't treat the plants with glyphosate. So after 3 generations, they have plants that have adapted to glyphosate and plants that have not had that adaptation. Then they did a treatment where they sprayed their Morning Glories with glyphosate, and they measured expression. And I should say this is only half of the big experiment they did, but this is the half that matters for this question. So they ended up had expression in plants that had evolved resistance to glyphosate. They had expression in control plants, and they had expression in control plants that had been sprayed with glyphosate. And so our thinking was that that purple plant up there, I'll give you these ones tell you about the evolved response to glyphosate, and these plants right here that have not experienced glyphosate before but have just been sprayed with them tell you about the plastic response to glyphosate. So we can compare our control plants to our plants that have been sprayed to get a sense of expression plasticity, and we can compare our control plants to the plants that evolved in response to glyphosate to get a sense of evolution. And then we can think about whether these align. When you spray a plant with pesticide, does the plant do what it might do when it actually evolved resistance? And if so, that would be consistent with adaptation. All right, so here's what the data looks like now. So each of these points is a different gene. On the x-axis, I'm showing the plastic response. So genes over here increased in expression in response to spray. Genes over here decreased in expression. The y-axis is the evolved